Good day, everyone. It's another day for another discovery.
Indeed it is. So welcome back to Life Discovery News TV. My name is Mark Adrian Aquino.
And I'm Akesha Irish de Guzman.
Bringing you
the Eye on the News for this day, Friday, December 10, 2018.
Microbiologist of the National Human Genome Research Institute is taking a step towards the future through studying cell division to find a cure for cancer. Let's take a closer look. Joanna Villa.
Good morning, everyone. We are here live at Baguio City in the Institute of the National Human Genome Research to have a word with the head of the microbiologist team. Good day, Ms. De Vera.
Good day, yes.
We have a few questions that we would like you to answer. What is your objective behind all of this?
As you can see, cancer is a leading cause of death worldwide. For that reason, many are afraid of this term due to the low rating of survivors. And people who have this easily lose hope. Well, some of them. So I wanted people to stop being afraid and give them hope that they can go through this by trying to create a cure.
This cure you are talking about, what is it?
Well, some parts are confidential, but it has something to do with human genetics, our cell division.
Upon mentioning cell division, what is its connection to the cancer disease?
This type of disease causes the cells to divide uncontrollably. It describes the results when cellular changes cause the uncontrolled growth and division of cells.
Can you provide further details, Ms. De Vera?
Okay, so a cell divides and reproduces in two ways, mitosis and meiosis. This is a process under the cell cycle that, as mentioned above, is known as the cell division. Mitosis and meiosis are the processes that have normal checks and balance in place that controls and limits cell division. And cancer cells are produced otherwise, having no normal checks and balances.
What inspired you to conduct this study?
It's what I do. It's basically my job.
That's all of my questions. Thank you, Ms. De Vera. This has been Joanna Alessavilla, live from Baguio City. Now, let's hear a word from Ara through Google Meet for further details.
Yes, Joanna, thank you. This is Jaisal Araparas reporting. Today, I am here with the microbiologist team scientist, Ms. Shane Mandal. A pleasant morning to everyone.
Pleasant morning to everyone.
According to the head of your team, ma'am, creating the cure for cancer has something to do with the division of cells. How and in what way?
As there is specifically in the stages and order of cell division, mitosis and meiosis falls into the cell cycle, which is responsible for preparing the cell division. Now, in order to understand how cells are able to produce cancerous cells, we shall start with the first type of cell division, the mitosis. Mitosis is a type of cell division that results in two identical daughter cells, and its major purpose is for growth and to replace worn-out cells for if not corrected in time. Mistakes made during this process can result in the changes in the DNA that can potentially lead to genetic diseases. Whereas this process is divided into 5 phases, starting with the interphase wherein the DNA in the cell is copied for preparation for cell division where this results in 2 identical full sets of chromosomes. And during this phase, microtubules extend from these centrosomes, then going to prophase in which chromosomes pair up where there is also the metaphase. The chromosomes line up neatly end-to-end along the center or equator of the cell. After that, the sister chromatids are then pulled apart by the mitotic spindle, which pulls one chromatid to one pole and the other chromatid to the opposite pole, where in this stage is recognized as the anaphase. Next to anaphase is the last stage of mitosis, the telophase, where at each pole of the cells, a full set of chromosomes gather together and the membrane forms around each set of chromosomes to create new nuclei. The single cell then pinches in the middle to form two separate daughter cells, each containing a full set of chromosomes within the nucleus, whereas this process is known as cytokinesis. As the cell reaches the fourth phase in mitosis and undergoing the cytokinesis process, its journey doesn't end there, for upon the succeeding the mitosis division, the meiosis division follows, which is the second type of cell division, the process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells are known as our sex cells and the sperm cells. Sex cells or sperm cell in male and egg cell in female. If the mitosis is divided into 4 stages, meiosis can be divided into 9. These are divided between the first time the cell divides are labeled as the meiosis 1 and the second time it divides are known as the meiosis 2. Beginning at the meiosis 1 is the interphase where the DNA in the cell is copied, resulting in 2 identical full sets of chromosomes. Then the prophase 1 wherein the copied chromosomes condense into X-shaped structure that can be easily seen under a microscope. Each chromosome is composed of 2 sister chromatids containing identical genetic information. The chromosome pair up so that both copies of chromosomes 1 are together, both copies of chromosomes 2 are together, and so on. The pairs of chromosomes may then exchange bits of DNA in a process called recombination or crossing over. At the end, prophase 1, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. The meiotic spindle, consisting of microtubules and other proteins, extend across the cell between the centriole. Following the prophase 1 is the metaphase 1, in which the chromosome pairs line up next to each other along the center or the equator of the cell. The centrioles are now at opposite poles of the cell, with the meiotic spindles extending from them, whereas meiotic spindle fibers are attached to one chromosome of each pair. Next to that is the anaphase 1. The pair of chromosomes are then pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and the other chromosome to the opposite pole. In meiosis 1, the sister chromatids stay together. This is different to what happens in mitosis and meiosis 2. Lastly but not the least, is the telophase 1 and cytokinesis. The chromosomes complete their move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather together. A membrane forms around each set of chromosomes to create 2 new nuclei. The single cell then pinches in the middle to form 2 separate daughter cells, each containing a full set of chromosomes within the nucleus. This process is known as cytokinesis. Afterwards, the meiosis 2 will come into the picture, where the cell will undergo the prophase 2, wherein there are now 2 daughter cells, each with 23 chromosomes or 23 pairs of chromatids. In each of the 2 daughter cells, the chromosomes condense again into visible X-shaped structures that can be easily seen under a microscope. The membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. The centrioles duplicate. The meiotic spindle forms again. As the meiotic spindle has been formed, the process in metaphase 2 will now begin. In each of the 2 daughter cells, the chromosomes, or pair of sister chromatids, line up end to end along the equator of the cell. The centrioles are now at the opposite poles in each of the daughter cells. Meiotic spindle fibers at each pole of the cell attached to each of the center or sister chromatids. After that, anaphase 2 will set about. The sister chromatids are then pulled to opposite poles due to the action of the meiotic spindles. The separated chromatids are now individual chromosomes. Then part of the completion of the cell is the telophase 2 and cytokinesis. The chromosomes complete their move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather together. A membrane forms around each set of chromosomes to create two new nuclei. This is the last phase of meiosis. However, cell division is not complete without another round of cytokinesis. Once cytokinesis is complete, there are four granddaughter cells, each with half a set of chromosomes or haploid. In males, these are four cells are all sperm cells. In females, one of the cells is an egg cell, while the other three are polar bodies, small cells that do not develop into eggs. Basically, if one of the stages in the process fail or an error occurs as cells are dividing, genes can lead to uncontrolled cell growth or potentially resulting in genetic mutations that can lead to a variety of diseases such as cancer, as cancer is an unchecked cell growth and the result of the unregulated process of the cell cycle due to the breakdown of the mechanism that controls the entire process.
That sure is a one long process. I'm sorry to interrupt, but how are you going to develop the cure?
I cannot say anything else apart from making a device that can alter to prevent the process from breakdown.
And what does it mean exactly?
Eliminating the cells gone wrong.
Is there anything else or you want to add?
So far, I have reached the limit of what I can disclose.
Well then, that fills all the spots. It is a pleasure to have this interview with you, Ms. Shane Mandal.
Well then, thank you for inviting me as well.
Again, this is Jaisal Ara Perez reporting, LDN TV, back to studio.
Thank you, Jaisal and Joanna.
Those were the top news for this hour. To those who are fighting some illness, specifically cancer, keep on fighting. Hope is on the way.
Yes, we hope everyone will have a great day ahead. And for more sizzling news of truth and accuracy, keep on watching Life Discovery News TV every morning. Once again, I am Akesha Irish de Guzman.
And I'm Mark Adrian Aquino,
Delivering news from worldwide. Have a great day.